The cell wall is really the most common drug target for most antibiotics, simply because the cell wall is one of the unique features that bacteria have, which our cells do not. And so again, the majority of antibiotics prescribed all around the world are called beta-lactam antibiotics. It has to do with their structure. So the beta-lactam is a ring structure found in penicillin and cephalosporin classes of drugs. And so these inhibit cell wall biosynthesis. So the idea would be, as a bacterial cell is trying to replicate and building its new cell wall, these beta-lactams bind to the proteins necessary to build the cell wall and prevent those proteins from building the cell wall. The first antibiotic really discovered and marketed in the US was penicillin, which has this mechanism. Now, one of the issues with antibiotic use, especially in the US and in some other, I guess what we would call more industrialized countries, is how much antibiotic usage occurs in our agriculture. So animals are given antibiotics because we know that when you give antibiotics to an animal, it can boost their growth. And of course, it can prevent infection. But these antibiotics are not given when the animal has an infection. They are simply given throughout the course of the animal's life. The issue with that is that constant exposure to antibiotics can, of course, lead to the development of resistance. So one of the major sources of antibiotic-resistant microorganisms is actually agricultural use of antibiotics. So 64% of our antibiotics go to agriculture, 36% go to people. The frequency of antibiotic usage, I told you that cephalosporins and penicillins are both beta-lactams. Beta-lactams are the most common types of antibiotics, and you can see that combined beta-lactams occur for about 64% of antibiotics used all around the world. Quinolones and macrolides can inhibit DNA or protein biosynthesis, and then other mechanisms of action make up another small percentage. Here they're showing you natural penicillin, which is called penicillin G. Penicillin is derived from a species of fungus the genus is called Penicillium. And so it has this natural structure. This is what it looks like. Here's the beta-lactam moiety. And what scientists have done since the discovery and isolation of penicillin is to modify the natural structure to allow for better activity and to allow it to be resistant to beta-lactamases and resistant to acids. So beta-lactamases are enzymes that some bacteria can produce to allow them to destroy the antibiotics. And so you see things like methicillin are supposed to be acid stable and beta-lactamase resistant. Oxacillin is supposed to be the same. However, things like ampicillin while it's acid stable, it is beta-lactamase sensitive. And then carbanicillin is also acid stable, but it is beta-lactamase sensitive. So these modifications of these drugs have been really important to allow better efficacy and, with the hopes of having less resistance. But of course, we all know that there is methicillin resistance because we are aware of methicillin-resistant Staphylococcus aureus. Protein synthesis is also an attractive drug target. While both bacteria and eukaryotic cells do protein synthesis, bacterial ribosomes are different from eukaryotic ribosomes. So many of these antibiotics that target protein synthesis will target features of the ribosome that are in the prokaryotic ribosome or the bacterial ribosome versus the eukaryotic ribosome. So again, our ribosomes are slightly different, and that's what gives that selective toxicity. These drugs should inhibit the bacterial ribosome before they inhibit the eukaryotic ribosome. Now, that's not to say that these drugs can't interfere with the eukaryotic ribosome. There is always that risk of off-target effects. However, preferentially, they target the bacterial ribosome. Some examples of antibiotics that target protein synthesis, they give you some classes here, including the aminoglycosides, tetracyclines, and macrolides. Nucleic acid synthesis is an attractive drug target because the enzymes that bacteria use to generate their DNA are slightly different from those that we use. So quinolones are a class of drug that interfere with DNA gyrase. So ciprofloxacin is one of those. Remember, DNA gyrase is necessary to unwind the supercoiled DNA so that it can be opened up and copied. Without the ability to do that, unfortunately, the bacteria will not be able to replicate their DNA. Other antibacterial drug targets can include biosynthetic pathways that perhaps eukaryotic cells don't have, things that will interfere with vitamins or amino acids or other types of compounds that the cell will try to incorporate but won't be useful for what the cell actually needs, which can lead to the cell's death. Sulfa drugs are an example of antibacterial compounds that fall into this category. For example, sulfa drugs are used to interfere with folate biosynthesis. We don't produce folate, eukaryotic cells, we don't produce folate. We get folate from our diet. It's one of the B vitamins, but bacteria don't pick up folate, they have to make it. So sulfa drugs can inhibit folate biosynthesis, which will lead to antimicrobial activity in the cell. So even though we make a big deal about how penicillin was, the first antibiotic on the market, it made a huge impact in how we were able to treat soldiers during World War II. And, I would argue helped us to be successful during World War II because we were able to better treat our soldiers. The Germans also did have antibiotics. They had sulfa drugs, which were discovered in the 1930s, but penicillin was just a little bit better. Interestingly enough, even though penicillin was described and published first, the discoverer of sulfa drugs and their antibacterial activity, Gerhard Domach, he actually won the Nobel Prize in 1939, whereas Alexander Fleming and his collaborators didn't win the Nobel Prize until 1945. Another type of antimicrobial drug is isoniazid. So this is useful really only in treating infections that are caused by acid-fast organisms because isoniazid interferes with the biosynthesis of the mycolic acid. So as long as a cell has mycolic acid, it should be sensitive to this compound. Daptomycin is also a relatively newer compound. It causes pores to form in the cytoplasmic membrane. This is useful for gram-positive infections. It would be less useful for gram-negative infections because remember, they have two membranes, so you'd have to be able to punch holes in both. This platincimicin is a new type of antibiotic. It has broad spectrum activity, especially against methicillin-resistant Staph aureus and vancomycin-resistant enterococci. This drug in particular targets some of the steps involved in the fatty acid biosynthesis of gram-positive cells. They use a slightly different fatty acid composition than some of the gram-negatives, and so it has shown efficacy against some of these otherwise antibiotic-resistant gram-positive infections like Staph aureus and the enterococci.